Signal Transduction by me and Brenda. Cells must communicate with their environments in order to react properly. Without a cellular response, cells could not adapt and would die under certain conditions. There are 3 steps that a cell must carry out in its response. Small molecules called ligands are attached to the protein's membrane to create a ligand-receptor complex with its receptor. And this causes a change in shape. Since structure fits function, the protein's function changes as well. This can increase the rate at which the cell can react because the cell won't need to spend time making additional proteins that would do the same job as the modified proteins. The activated protein transmit a signal sequence through a cascade of proteins known as the signal transduction pathway. The signal can then reach the nucleus, where the cell can form a response. As mentioned earlier, a cell needs to adapt to its environment, but it also needs to be able to communicate with other cells to allow passing of materials, the formation of neuron connections, the prevention of cell density, and many other vital system functions. Examples of the reactions to the signal are also shown here. In addition, these pictures have been provided. This first picture has a general overview of the process of cellular response. The second picture goes into further detail, illustrating the process with a specific ligand, growth factor. The protein cascade is shown, and the change in the transcription factor is also shown. The situation. Tessa is emailing her friend Melanie, who is a doctor, about her champion dog's erectile dysfunction. Tessa bought the dog for breeding, and ED inhibits an erection during sexual activity. This is obviously a problem. Erection is controlled by a cell signaling pathway, which is illustrated in the email that Melody sends to Tessa. Melody suggests Viagra and ginseng, whose effect is explained in another email that's sent to Tessa. The first part of the assignment calls for us to name the first and second messengers as well as the cellular response that follows the process. Then we must explain the significance of the arrow's size. Finally, we'll examine the receptor's location. First, let's explain what's happening in the picture in email 1. This picture explains the process that causes an erection via cellular response. First, nitric oxide can be identified as the ligand, and therefore the first messenger that starts signal transduction. It's made from the amino acid L-arginine, bound to the nitric oxide synthase enzyme, which is found in endothelial or neuronal cells. Also bound to this enzyme are calcium and CAM. Nitric oxide is then produced along with citrulline, which does not take part in causing an erection. Since nitric oxide is the ligand, it must bind to its receptor in the smooth muscle cell, which is, in this case, guanylyl cyclase. This enzyme catalyzes the reaction that turns GTP into cyclic GMP, or cGMP for short. This is the second messenger, which activates PKG. And the cyclic guanylyl monophosphate-dependent protein kinase. That's PKG. And a kinase is an enzyme that transfers phosphates. The proteins in the signal transduction pathway are phosphorylated by the PKG using ATP, becoming phosphoproteins. And then this causes the calcium levels to decrease, and that relaxes the smooth muscle tissues. Since the muscles relax, it can allow for a better blood flow into the penis, causing an erection. So we know now that the nitric oxide binds to the receptor protein guanylyl cyclase to make it active. You can look at the arrow here between these two, and it's small because it's a single movement of substrate to enzyme. After this happens, the arrow size in the picture is increased for each reaction. These large arrows indicate the amplification of the products formed. Recall the meaning of cyclase. It's the protein that enables the cell's reactions to continuously occur, to continue the cycle by leading to the activation of the necessary enzyme, since it is a kinase that transfers phosphates, PKG. So, the cyclase can affect many proteins, whereas nitric oxide affected only the cyclase by turning it on. Still, this small arrow cannot be ignored. Without the nitric oxide to turn on the cyclase, the other reactions would not occur. So now we can discuss whether guanylyl cyclase is located on the cell's plasma membrane or intracellular membrane. If we examine nitric oxide, we can see that it is small and nonpolar and that it could easily diffuse across the membrane. Therefore, the molecule would travel into the cell where it could bind with guanylyl cyclase located on the intracellular membrane. Let's move on to the second assignment. First, we'll explain the expected effect of ginseng on nitric oxide and why. Then we'll talk about Viagra's expected effect on phosphodiesterase. Finally, we'll talk about the protein that we would expect to be broken if neither of these chemicals work. This picture from email 2 is similar to the previous one, though this one is obviously a more detailed diagram. We can still recognize the nitric oxide and guanylyl cyclase, as well as the GMP and calcium, relaxation, and also PKG. However, the figure becomes more complicated. We're introduced to phosphodiesterase, and that's shown here as PDE5. This enzyme inhibits the cGMP, and that causes the cellular response of erection to stop. Because cGMP is continuing the process, if you break the enzyme, the whole process won't work. It makes sense. Then there are PDE5 inhibitors which, true to their name, inhibit PDE5 to prevent cGMP to be broken down by PDE5. And an example of such an inhibitor would be Viagra. PKG is activated by cGMP. And we know this from the previous diagram. One of PKG's functions is to phosphorylate a protein called lambin in phospholambin, or P-lambin, as it is shown. It's important that this occurs because if left unphosphorylated, lambin would inhibit protein pumps so that calcium would build up and make the muscle contract. And this is the opposite of what we're trying to achieve. We want calcium to be released, which is why the phosphorylated lambin's activating protein pumps is good. Northeast of PKG, we can spot the IP3 receptor in the diagram, also known as iocytyl triphosphate. These receptors help in opening calcium channels that let calcium flow into the cell's cytoplasm. Since our objective is to decrease the amount of calcium in the cell and yield the relaxation of muscles, activated IP3 receptors will hinder the process, and for this reason, PKG inhibits IB3 receptors. MLC phosphatase, an enzyme that removes phosphates from proteins, is also activated by PKG. This enzyme catalyzes the change of MLC, the myosin light chain, to MLCP, which is the phosphorylated myosin light chain. MLCK is the myosin light chain kinase that has the opposite function of MLC phosphatase. It gives phosphates to proteins instead. As the picture clearly shows, the enzyme would cause contraction. Therefore, we would not want to activate this enzyme, but rather MLC, which causes relaxation. The ginseng would allow for an increase in nitric oxide. This can be expected since the only effect a substance would have on nitric oxide is to decrease or increase its presence in the cycle, and we know that ginseng is supposed to enhance erection. This increase is good for the signal transduction process since more nitric oxides would activate more guanylyl cyclases to activate more cyclic GMP, which would lead to more calcium release and more muscle relaxation. Therefore, by adding the amount of nitric oxide present, ginseng amplifies the process further to enhance erection. It is expected that Viagra inhibits PDE5, or phosphodiesterase, and this is because PDE5 breaks down the cyclic GMP that allows the processes in the cell to occur. Thus, inhibiting the activation of PDE5 will allow for cGMP to continue functioning since it is not being broken down. And this would eventually lead to the necessary calcium release needed to relax the muscles, explaining why Viagra helps males to get an erection. Yeah, sorry for the delayed animation. Anyway. If neither ginseng or Viagra worked in getting Tessa's dog an erection, a protein in signal transduction would be broken, and the suspected protein would be PKG. You can kind of see it's freaking out right now. The cyclic guanylyl monophosphate dependent protein kinase is responsible for several functions that lead to an erection. With a broken PKG, the unphosphorylated lambin would inhibit protein pumps, leading to a buildup of calcium. In addition to this, calcium channels would remain open due to an activated IP3 receptor, greatly increasing calcium concentration in the cytoplasm. A broken PKG would also fail to activate MLC phosphatase, which causes smooth muscle reaction. Well, that's the end. I hope you enjoyed, and I hope you learned something.